Let's take a look at the phases of meiosis one by one. Remember, interphase, the cell is going to undergo the G1, the S phase, and the G2. You know that this is the same whether you are in mitosis or in meiosis. Now, let's assume that S phase has occurred. The 46 chromosomes, each of them has been copied, right? Which means that the sister chromatids are held by centromeres, right? Now, what happens during the first prophase? Meiosis 1. So in meiosis 1, during prophase 1, what happens, as expected, just like in mitosis, the nuclear envelope is going to be fragmented, okay? Nothing special there. Then, what are we going to see? We're going to see homologous chromosome pairing up together, okay? Synapsis develops. Then, we are also going to see a crossing over developing between homologous chromosomes, right? The spindle are forming, and the spindle are nothing but microtubules that are going to grow out, right, toward the kinetochores, okay? The centrioles are going to move toward opposite direction, okay? Until these spindles attach to kinetochores. Only the kinetochores that are visible are going to see some attachment of microtubules on them, right? So that because of the formation of synapses, some kinetochores are going to be hidden. But don't worry about that. I'm not going to bug you with that detail. What I want you to know is that the two homologous chromosomes, one is going to receive actually one spindle attaching to one of the homologous chromosomes, and the other spindle is going to attach to the other homologous chromosome. They are going to pull toward opposite direction. And as they pull toward opposite direction, this is going to force the homologous chromosomes to sit on the equatorial line. We are now in metaphase 1. All right? And now, as the spindles keep shrinking toward opposite direction, this is going to pull the homologous chromosomes away from each other, so they will keep migrating toward opposite direction, right? That migration is going to cause the chromosomes to end up in two future daughter cells. We are in anaphase 1. Eventually, in telophase 1 / cytokinesis, what are we going to see? We're going to see a cleavage furrow developing, okay? Now, you see that one set of the chromosome in a homologous pair is going to end up in one cell, and one set in another cell, okay? As consequence of that, each of this daughter cell, is haploid, right? Now, just like in mitosis, the nucleus is going to form around this set of chromosomes, so that you have two new nuclei that are going to form, and two daughter cells are going to be completely separated, right? We have now haploid cells, and these haploid cells are going to be subject to another cell division. And at that point, the second cell division is now going to focus on the fate of the sister chromatids, right? So now we are entering meiosis 2. In meiosis 2, we are going to have prophase 2. During prophase 2, the centrioles are going to duplicate and move toward opposite direction. That one. The nuclear envelope is going to get disorganized. That two, right? The spindles are going to develop Okay? And these spindles are going to target the kinetochores on each duplicated chromosome. Okay? So now the spindles being attached to the to the kinetochores, okay, this are going to be pulled toward opposite direction, okay, going to be pulled toward opposite direction. And now we are in metaphase 2, the middle, sit in the middle, okay. And from metaphase 2, as the spindle keeps shrinking, we go to anaphase 2, okay. Now the sister chromatids are now separated. They are migrating toward opposite direction, and they are getting ready to end up into two separate cells. Eventually, after anaphase 2, we are going to have telophase 2, where we're going to have, again, cleavage furrows, right? We are going to have the nuclear envelope that is going to reconstitute around the chromosomes. And so that we are going to have 4 daughter cells that are all going to be haploid. Right? Now, not only these cells are haploid, but very importantly, these cells are genetically different. Again, these cells are genetically different. Very important to remember. Now, in terms of meiosis in males and in females, for the formation of sperms, all sperm cells have about the same size. But that equality of size in the 4 haploid cells that are produced as consequence of meiosis in males is very different when we look at the female. In the female, the story is different. The egg actually does not split equally the cytoplasm among daughter cells. No. The eggs are actually, the only thing they're going to split equally is going to be the genetic material, right? But one cell is going to retain the vast majority of the cytoplasm, while the other cell is only going to retain the genetic information and a very small fraction of the cytoplasm, so that you have a huge cell and a teeny tiny cell that we actually call a polar body. Okay, now this is not a reproduction class, so I'm not going to dive into the details of that, and even what you call the different stages of oogenesis. I'm not going to dive into that. But if you are curious to know about that, I can always address that in another context. Anyway, now let's compare mitosis and meiosis. When we compare mitosis and meiosis, now during the first division of meiosis, we're going to have prophase 1, metaphase 1. Now, when we look at what is happening in prophase 1 and metaphase 1, it's very different from what is happening in prophase and metaphase during mitosis. The only similarity is that the genetic information is going to be condensed, right? In both cases, right? The genetic information is going to be condensed. The spindles are going to attach to kinetochores. However, here in meiosis, they don't attach to literally all the kinetochores, because not all kinetochores are visible in meiosis 1, while in mitosis all kinetochores are visible. Therefore, this one guy can receive one kinetochore, and I'm sorry, a spindle, and this one can receive a spindle, and that's how you see all the chromosomes that are lined up. While here, well, the homologous chromosomes are sitting on opposite direction, right? The second thing that is very importantly different, it's that during meiosis, one, you have in prophase crossing over that you never have in mitosis. Okay, from that perspective, they are very different, right? Okay, mitosis, we are focusing on sister chromatids. Meiosis one, we are focusing on homologous chromosomes, and then we go to anaphase one. Anaphase one and telophase one. So here we have anaphase one, right? And here we have telophase one that is going to produce two brand new daughter cells, while here we're going to have anaphase and eventually telophase with the cleavage furrow here to produce daughter cells here that are 100% genetically identical, containing twice the number of chromosomes, which means that these cells are diploid cells. They are identical genetically, while in meiosis 2, we're going to have the cells that are haploid, haploid and genetically different, right? We have 4 daughter cells here in meiosis, while in mitosis, we have 2 daughter cells. So that's a comparison between meiosis and mitosis. Now, during the process of meiosis, what we notice is that there are some instances where when the genetic information is not equally distributed among the daughter cells. This can happen during meiosis 1. It can happen during meiosis 2. Now, the outcome is the consequences are more drastic when it happens during meiosis 1 than when it happens during meiosis 2. Okay, let's address that a minute. So non-disjunction, it's basically that homologous chromosomes or sister chromatids fail to separate during the nuclear division, right? This causes unequal distribution of the chromosomes. Now, let's take a look at the non-disjunction occurring in meiosis 1. Say here we are in meiosis 1 and we are tracking 2 homologous chromosomes, right? We have non-disjunction there so that you have unequal distribution of chromosomes during meiosis 1, okay? So one cell is going to end up with less genetic information. Another cell is going to end up with too much genetic information. So the 2 cells are abnormal. Now, after meiosis 2, okay, the 4 cells are abnormal, right? 2 lacking or missing a chromosome, 2 having extra chromosomes, right? We call this aneuploidy. I will not bug you with that, but let me write it for you anyway. Aneuploidy. And aneuploidy means you can be actually missing 1 chromosome cell, n-1, okay? That's monosomy, case of monosomy. So monosomy, okay? Meaning that you are missing 1 chromosome, you only have 1 chromosome among the homologous chromosomes. And you can have n+1. This is a case of trisomy. Trisomy. You've heard, for instance, of Down syndrome, right? Okay, so now we have an extra chromosome. Okay, so all the cells are abnormal when non-disjunction occur during meiosis 1. That's a very bad news. Now, if the first division is normal, okay, so meiosis 1 happens normally, so this chromosome end up in one cell, and this other chromosome end up in one cell. So one do you receive from your dad, and this you receive from your mom, right? Now, let's take a look at if the non-disjunction occurs during meiosis 2. If that happens during meiosis 2, say non-disjunction occurs on this side, right? So we're going to have, this one is going to have n minus 1. That's monosomy, right? So going to have here n plus 1. This is trisomy, right? And here we're going to have n chromosomes that's normal and n chromosome that's also normal. So 2 gametes are normal while 2 are abnormal. All right? So we do see this non-disjunction occurring very much as people age. And this is particularly true for ladies producing eggs that have non-disjunction. That's why when a lady reaches 35 years old and above and she's conceiving, actually that pregnancy is considered a high-risk pregnancy. Why? Because we notice that chances of having extra chromosomes in an egg after a lady reaches 35 years old actually increases tremendously, right? When we look at the graph in the chances of having a child with trisomy 21, right? So this is age 35 years old. And here, the chances of trisomy, okay? So here, trisomy. Okay, so the chances increase tremendously after the age of 35 years old. Okay, all right. Okay, so here we have an example of non-disjunction where we do have in here an individual on this karyotype shows actually not 46 chromosomes but 47 chromosomes with an extra chromosome here, okay, in chromosome number 21. All right, so now some defects associated with non-disjunction. We have the Turner syndrome case. In the Turner syndrome case, this typically is going to be for individuals who are XO. In other words, this individual have an X chromosome, but they don't have another X or a Y chromosome. By default, these individuals are female. These female are going to have a webbing of the neck. They're going to have a short stature. They're going to be sexually underdeveloped, okay? Underdeveloped sexually. Sometimes ladies with Turner syndrome may actually reach the age of adulthood and never actually experience menstruation, okay? They're going to have some pigments across the body. So these are some of the features that are characteristic to individuals with Turner syndrome. They are all female. Then other types of non-disjunction, we have the Klinefelter syndrome. In the Klinefelter syndrome, the individual is double X and a Y. Now, because the individual is Y, that individual is definitely a male, right? However, that male is going to, because of the presence of the two X's, is going to have features that are very much developed toward a female, with the male features that are going to be very small compared to the norm, right? They're going to be very small, and all the features are going to be very much going toward female features. Now, in terms of the bones, the likelihood or the frequency at which they are having osteoporosis is very high. You know, the breasts are going to be very developed, okay, compared to a male who has only one X and one Y. Okay. What else should I say here? Oh, long legs and long arms. Okay. The arms and legs are going to be unusually long. The individuals are going to be taller than average. All right. How about poly-X? Poly-X individuals are going to occur in two kinds. We have poly-X that express or rather have triple X and poly-X that have four X's. The triple X individuals are definitely females, right? They are normal female. They have nothing special in terms of you know, their development. However, they tend to have exaggerated female features, like very much a high level of size of breast. They tend to be taller. They tend to be taller than average females, but otherwise they don't have any issue. You cannot tell that this individual here has this individual has triple X. And then you have the Jacob syndrome. The Jacob syndrome is cases where individuals are going to be X, Y, Y. This individual are going to be in general tall, with persistence, acne, and sometimes speech issues and reading problems. Let's take a look at the summary of this chapter. Now, as we compare the events that occur in meiosis and events that occur in mitosis, in meiosis 1 and mitosis, we are both going to have prophase. We already saw that. We're going to have in prophase 1 pairing of homologous chromosomes with crossing over, while in mitosis we do not have pairing of homologous chromosomes. There is no crossing over. In metaphase 1, we're going to have homologous chromosomes that are going to line up on the equator line, while we do have chromosomes lining up in metaphase during mitosis. However, that lineup is not dependent on being homologous chromosomes or not, right? Then we have anaphase. Anaphase 1 for meiosis 1 and anaphase in mitosis. So homologous chromosomes are going to migrate toward opposite direction, causing the cells to become haploid, while here for mitosis, the sister chromatids are the one that are going to separate into two different opposite direction. Okay, and then telophase 1 and telophase of mitosis. So telophase 1 and meiosis 1, we're going to have two haploid cells, while telophase in mitosis, we're going to have two diploid daughter cells that are genetically identical. In meiosis 2, during prophase 2, we have no pairing of chromosomes, and also in mitosis we have no pairing of chromosomes. So that's very similar, right? Metaphase 2, we do have haploid number of chromosomes that line up on the equator line, with each of the kinetochore with spindles attached to them, right? Here we have diploid. For mitosis, we have diploid number of chromosomes with spindle attached to each kinetochore, right? In anaphase 1, the sister chromatids are going to separate. In anaphase 2, the sister chromatids are going to separate, while in anaphase of mitosis, the sister chromatids are also going to separate and part ways. And then we have telophase 2 of meiosis 2, We have 4 haploid daughter cells that are genetically different, right? And different among themselves and sometimes among from the parents, right? Well, not sometimes. Actually, they are different from the parents, I should say. And while in telophase of mitosis, we have 2 diploid cells that are identical to the parent cells, right? And among themselves too, they are identical to the parent cells and also identical among themselves, okay? And finally, but not least, uh, do a review of this chapter before you move to something else. Look at the end of the chapter. You are going to see a review there, or you can design your own review of the chapter. Assess your understanding of the material. And as you assess your understanding of the material, write down some questions that you are producing yourself, right? By producing those questions yourself, you are really giving the chance to actually analyze the material and go through this material. Talk to your classmates. Do some questions at the very end of the chapter in your textbook, right? Finally, but not least, and like always, create a handwritten concept map that can be very helpful. Thank you for your attention. Bye now.